Okay, so in this lessons, we're going to talk about facilitated diffusion, which is a form of diffusion. So means that the general idea will be similar to the diffusions, means that they have to follow or down their concentration gradient. Okay, so in this case, against their passive process, they do not require ATP. So facilitated diffusions, we do know that phospholipids bilayer impermeable to water-soluble polar molecule, charged particle, and also the large molecule. For example, small protein, okay, for facilitated diffusion. Okay, so now for large molecule, a bit difficult for us to do the facilitated diffusion, but these two, we can actually increase the membrane permeability. Okay, for this charged particle on water-soluble particle to pass through, we simply provide a transport protein for them. Okay, we can provide the transport protein for them so that diffusion can take place. But to slightly differentiate between the normal diffusions and this kind of diffusion, so this kind of diffusion, we term it as facilitated diffusion. Means that with the help of what? Facilitate, right? Someone help them. Facilitate by who? By these transport proteins. Okay, and we do know that transport proteins, they are highly specific. So means that they don't share. Okay, they are highly specific. It's something like our key, right? The door key. Okay, we don't share the key. I don't use my key, okay, to open your door. Okay, similar in concept here, they are highly specific. So means that the ion channel that allow calcium ions to pass through, you don't expect potassium ion can pass through, even though both of them, they are positively charged. Are you clear? Okay, even sodium and potassium, they don't share their ion channel. Okay, so they are highly specific. And these transport proteins, we can differentiate them into two kinds of the transport protein. One is called channel protein or pore protein. Another one is the carrier protein. So we need to learn to differentiate between these two kinds of protein. Okay, for channel protein or pore protein, from the name is very, very simple. They do have a channel, or do they do have this, okay, the pore allow particle to pass through. So this kind of the channel generally for ion, small ions, no problem. Are you clear? Small ions, no problem. Water molecule, yes, they use this. Okay, as long as they are small, generally they can use this, what we call the channel proteins, right? If the something is very large and you give them channel protein, means that the pore size is huge, then other small particles also can pass through. You try to imagine if for glucose with a molecular weight 180, if you give them a channel protein, what will happen? So big, the hole, I mean the pore, anything also can pass through already, it become leakage. Are you clear? So for large particle, for example, glucose, amino acid, then we're going to use a carrier protein. Okay, so what's the difference between these two? Generally, channel protein or pore protein, the shape is fixed. They don't change the shape. They provide a channel for them to pass through. Can you see that? So if you look at my hand here, so it's a channel, so pass through. Can you see that? The channel to pass through. So they don't change the shape. But carrier proteins, they have to change the shape. If the carrier protein behave like the ion channel or pore protein, everything also can pass through, right? So they have to alternate between two precise three-dimensional shape. So you look at okay, how I do it. Okay, you can see this, right? My camera there. Okay, so you can see that this is something like this. They do. So I take it in, I change. Can you see that I alternate my finger, my hand? Right. So this alternative, the alternates between the two shapes allow us to bring the particle in and out. Okay, and highlights here, guys. So this particle, for them to move in and out, they need to have a binding site. So this one, write down, guys, in your notes. This site is termed as the binding site, and they are very, very specific. Are you clear? Binding sites. So it means that in the binding sites for glucose, you don't expect maltose can bind. Okay, you don't expect the galactose can bind. you don't expect that fructose can bind. Are you clear? So they do have this binding site. So this binding site is very, very specific. Okay. So for example, for channel protein, we have the ion channel. For example, sodium ion channel. You can write down another one, water channel. Water channel have another name called aquaporine. Sorry, aquaporine. Okay. You can write down another example, ion channel, as well as the water channel. Water channel also termed as aquaporine. So what is the mechanism? So the channel protein provide a water-filled pore or hydrophilic pore. So they allow the charged particle, usually ions, to move through the membranes. Okay. How about carrier protein? For example, we have glucose transporter, GLUT. GLU stand for glucose, T stand for transport. So it's a glucose transporter to transport the glucose. So what they do? So the molecule bind to the proteins. Which part they bind? They bind to the binding sites of the protein. Then the carrier protein can flip or alternate between two shapes. As a result, the binding site is alternately open to one side and closed at another side. So allow the particle to be brought in, okay, or to be transported in, okay, from regions to another regions. Are you clear? So in this case, because of the facilitated diffusions, therefore from high to low. Okay. Can? Okay. Now, let us look at another special kinds of the ion channel. This ion channel is called gated channel. They have the gate. So something have to cause them to open. So stimulus in this case will cause them to open. So initially, can you see that they are closed? So no matter how high the concentrations of the ion outside compared to inside, they cannot diffuse across. Are you clear? But if you have the certain messenger, as a hormone, neurotransmitter, certain chemical can bind and cause them to open, therefore the facilitated diffusion can take place. So this one can act as something like a control. Okay, something like a control allow us to actually control the times where we want the facilitated diffusion to take place. For example, if you have the hormone, when the release of the hormone only cause the ions to move in or out. In this case, can we control directions? No. Generally, I say that no, you cannot control directions. So this direction will depend on the concentration gradient, which part actually have higher concentrations of the particle compared to another part. If let's say outside is high, inside is low, then they will move from the tissue fluids into the cells, okay, until they reach the dynamic equilibrium. Okay, so the rate of the facilitated diffusion totally depends, as I say, that total surface area. It's the steepness of the concentration gradient. Guys, put it in. I prefer the word steepness. Okay, of the concentration gradient. Now, they also depend on the temperature. Correct or not? Okay, and for this one must be water soluble. So that's nothing to do with the nature of the particle, but generally they depends on the number. So surface area is not really very, very important. If try to imagine if this is my surface area, both of them have the same surface area, but the first one only have 2 ion channel. The second one have 4 ion channel. So therefore, surface area same, but the number of ion channel is more, 2 compared to 4. So this one will have higher rate compared to the surface area for A. Are you clear? Okay. And also, if these ion channel, they are gated one, then we have to look at it whether they open or not. Okay, no point. I have a lot, but cannot use one. Are you clear? Okay, I have 10 car. What kind of car? Or toy car. We cannot fetch people. Useless. Okay, so this is why I say that's the number. Okay, the number important also depend whether they opens or not. If they are not opens, then useless. Particle cannot pass through. Okay, so with this, I have done for the lesson. So today,